This video will provide you with an overview of the nervous system, including its development. In addition, we'll also look at the contents of spinal nerves. In this video, I first want to expose you to some embryology in order to lay the groundwork for the remainder of today's discussion on the nervous system. Let me first give you the backstory that leads up to the image on the screen. By this point in development, the embryo has implanted into the wall the uterus. The extra embryonic structures that help to support the embryo have also developed. For example, the amniotic membrane that surrounds the amniotic cavity has developed, and the yolk sac has developed that provides nourishment to the embryo until the umbilical vessels form. By week 2, or day 14, the embryo has taken the shape of a trilaminar disc. The 3 layers of this disc are responsible for developing into various tissue types. The ectoderm, shown here in blue, gives rise to the central nervous system. In addition, the integumentary system, including sweat glands, also develop from the ectoderm. The mesoderm, which is the middle layer, gives rise to the muscles, bones, connective tissues, as well as gonads and kidneys. The innermost layer of the trilaminar disc is called the endoderm, colored here in yellow. The endoderm differentiates into cells that line the GI tract, the respiratory system, and the genitourinary system. As further development and proliferation of the cells within the trilaminar disc continues, there comes to be a very critical and a very active stage during weeks 3 and 4 when the embryo's nervous system takes shape. The embryonic process by which the early precursors of the central nervous system are formed, from the ectoderm of course, is referred to as neurulation. Figure A is an illustration of a 21-day-old embryo that is roughly 1 millimeter in length. That's about the size of a pinhead on a sewing needle. Located along the central axis of the embryo is the notochord. During embryonic development, the notochord is the signaling center that helps to orchestrate the timing of tissue development and differentiation. Once the notochord has fulfilled its job as the signaling center, as the vertebral column develops, the notochord becomes incorporated into the intervertebral discs as the nucleus pulposus. Signals initiated by the notochord cause the ectoderm, shown in these images in blue, to thicken, which forms a neural plate. These are the neural plates. These neural plates form 2 lateral folds called neural folds, which are separated by a shallow furrow called the neural groove. In figure C, the neural folds protrude upward and begin to grow toward one another until they unite and fuse, forming the neural tube. From this point forward in development, the neural tube will be responsible for giving rise to the brain and the spinal cord. Also take note of the neural crest cells, shown in purple throughout, which give rise to a host of different derivatives. Because the neural crest cells are adjacent to the neural tube, it's probably no surprise that they differentiate and migrate laterally to form the peripheral nerves, which include cranial nerves, spinal nerves, and ganglia. Here is where the neural crest cells reside after the neural tube is developed. On occasion, the folding of the neural tube can go awry, leading to congenital neural tube defects. Abnormal closure of the neural folds can involve the meninges, vertebrae, and even overlying muscles and skin. Neural tube defects are said to occur in about 1 out of every 1,000 births. It has been estimated that about 70% of all neural tube defects can be prevented if expecting mothers take their prenatal vitamins with folic acid prior to and during pregnancy. Spina bifida occulta is a condition in which the vertebral arch fails to close posteriorly. In its most minor form, the only evidence of spina bifida occulta may be a small dimple with a tuft of hair in the lower back. An overlying lipoma, dermal sinus, or birthmark may also occur. Spina bifida occulta usually produces no symptoms, and only a small percentage of affected infants have neurological defects. Spina bifida cystica is an umbrella term that describes a cyst protruding through a defect in the vertebral column. When the cyst only contains meninges and cerebral spinal fluid, the defect is called spina bifida with a meningocele. While the spinal cord and spinal roots lie in their normal position, there may still be spinal cord defects. If the spinal cord and/or nerve roots are contained within the cyst, the defect is called spina bifida with a myelomeningocele. Myelomeningoceles are more common and more severe than spina bifida with meningocele alone. While myelomeningoceles can occur anywhere along the vertebral column, they are most common in the lumbar and sacral regions. Myeloschisis is the most severe form of spina bifida. In this defect, the spinal cord in the affected area is open to the external environment because the neural folds fail to fuse. The word schisis means cleaving. As a result, the spinal cord is represented by a flattened mass of exposed nervous tissue. This condition usually results in permanent paralysis of the lower limbs. The little girl on the left of the screen suffers from spina bifida occulta, as evidenced by the dark tuft of hair on her lower back. The second image is of an infant with spina bifida cystica. To determine if this is a meningocele or myelomeningocele, imaging with an ultrasound, for example, would need to be done. With ultrasound resolution now being the best it's ever been, many of these birth defects can be identified in utero. Early identification prepares both the families and the physicians for what to expect after birth. During the formation of the neural tube, somites are also formed from the paraxial mesoderm, which is tissue that flanks the notochord, shown here. Here's the notochord. Somites are a population of cells that give rise to a variety of important structures, including skeletal muscle, cartilage, tendon, and vertebrae. And they also form the dermatomes of the skin. More specifically, somites specify the placement of the spinal nerves, and they guide the nerves on their course to innervate the muscles and skin derived from the same somatic levels. So the peripheral nerves that are formed from the neural crest cells are guided by the somites to their terminal effector tissues. A little further along in development, as the torso and the limb buds begin to take shape, we can now appreciate how the muscles derived from a single somatic level are innervated by the spinal nerve associated with that somite. In this image, we're seeing an epimere, which is the dorsal portion of a somite, forming the muscles that will extend the back, so the erector spinae muscles, for example. Notice how those erector spinae muscles are innervated by dorsal primary rami. Again, the somites are guiding these nerves to their effector tissues. Also recognize that a myotome is a specific part of a somite that develops into a group of muscles that are innervated by a single spinal nerve. However, a single muscle may be formed from multiple myotomes. Therefore, you'll soon learn that muscles are innervated by more than one spinal nerve level. Let's look at an example to better illustrate this point. Let's take the deltoid muscle as our example. The deltoid muscle, which overlies the shoulder, is innervated by the axillary nerve, which comes from spinal cord levels C5 and C6. Therefore, we say that the deltoid is formed from myotomes from cervical somites C5 and C6. Not only are nerves migrating to innervate the muscles of the limbs, but they are also migrating to innervate the skin. The distribution of a single spinal nerve that innervates a localized area of skin is called a dermatome. Imagine that you received a flu shot here in the arm. The needle prick that you feel from the flu shot would be conveyed back to the central nervous system via the C5 spinal nerve. This particular area is referred to as the deltoid skin patch. So why, as a physician, are dermatomes important? Well, they can actually be very useful when trying to identify the location of a nerve lesion, for example. As we progress along, you'll see dermatomes come up again and again. For the purposes of this block, you will need to know the dermatomal distributions along the back, the upper limbs, and the lower limbs. Knowing these distributions will help you to solve clinically related problems. This next section is likely to be a review from your basic biology course. The nervous system is divided into two parts. The central nervous system is derived from the neural tube and is comprised of the brain and the spinal cord, shown here in green. The peripheral nervous system is composed of all nervous tissue structures outside of the central nervous system. This system will connect the body back to the central nervous system, and structures of the peripheral nervous system shown here in purple include cranial nerves, spinal nerves, autonomic nerves, ganglia, and receptors. By definition, ganglia are collections of neuronal cell bodies and processes with a common function. An example would be the dorsal root ganglion. Cranial nerves are nerves which emerge from either the brain or the brainstem. And lastly, spinal nerves exit between vertebrae through the intervertebral foramina. Not only can the peripheral nervous system be divided into spinal nerves and cranial nerves, but it can also be divided functionally into somatic and autonomic nerves. Somatic nerves innervate skeletal muscle and skin, while the autonomic nerves innervate the viscera and glands. To fully understand the story of spinal nerves requires that we start with some foundational concepts that you've probably seen before. Recognize that the neuron is the functional unit of the nervous system, which is responsible for conveying electrical signals throughout the body. And neurons consist of three main parts. The cell body is going to contain the nucleus as well as a number of other cell organelles, and it's going to serve as the main hub of the cell. The dendrites are elongated processes that extend from the cell body and are specialized to receive stimuli from neighboring neurons at an area called a synapse, which is the junction between the two neurons. The axon is a single long process that ends as axon terminals, which are specialized for conducting nerve impulses to neighboring neurons or target tissues. The long, slender axon is often encased by a myelin sheath that is made from Schwann cells. Myelin helps to increase the speed and propagation of the nerve impulse. Just like the insulation on an electrical cable increases the speed and effectiveness of electron conduction, so too does the insulation increase the speed of electrical impulses through the nerve and through the neuron. The small gaps between the Schwann cells are referred to as the nodes of Ranvier. Within the peripheral nervous system, neurons are either motor or sensory neurons. Motor neurons are also called efferent neurons and are typically multipolar in morphology, just like the neuron described on the previous slide. When dendrites receive the signal from neighboring neurons, the impulses are conveyed through the cell body, and from there they are conveyed through the axon to the effector tissue, in this case, skeletal muscle. Recognize that efferent neurons are always conveying impulses away from the central nervous system towards an effector tissue, such as skeletal muscle. The somatic nervous system relies on a single neuron chain system to convey information from the spinal cord to the tissue of interest. That means that the cell body and the dendrites of these neurons are contained within the spinal cord. This means that axons of the somatic nervous system can be very long. Imagine an axon going from the spinal cord all the way down to the great toe. This could be about 3 feet in length, if not more. Sensory or afferent neurons have a pseudo-unipolar morphology. A long peripheral process receives and transmits the signals from the periphery to the cell body. Here's the long peripheral process. Here's the cell body. Recognize that these cell bodies are housed within the dorsal root ganglion. I'll abbreviate that DRG. From the cell body of the neuron, the neuronal impulse is directed to the central nervous system through a central process. At the end of the central process are synaptic endings, which communicate with the neurons in the spinal cord. Sensory neurons can convey different types of information. Exteroceptors convey information about pain, vibration, pressure, and temperature. Proprioceptive neurons convey positional information from muscles and joints. This positional information allows you to know at all times where your body is in space. Now let's take a look to understand how these neurons are organized to form functional nerves. Here's an overview of the gross anatomy of both the spinal cord and spinal nerves. The spinal cord itself is made up of both white matter and gray matter. The white matter consists mostly of myelinated axons that carry impulses to and from the brain. White matter also contains glial cells that support those axons. Gray matter, on the other hand, consists mostly of neuronal cell bodies. For example, the ventral horn of the spinal cord is made up mostly of cell bodies of motor neurons. In specific regions of the spinal cord, you'll also find lateral horns of gray matter, which house the cell bodies of autonomic neurons. Coming out of the spinal cord are the ventral rootlets, which converge to form the ventral root. And going into the spinal cord posteriorly are the dorsal rootlets, which are coming from a dorsal root, which is associated with the dorsal root ganglion, also known as the spinal ganglion. Again, the dorsal root ganglia are where the cell bodies are housed for the sensory neurons. When the dorsal and ventral roots converge, they form a single spinal nerve. The spinal nerves are relatively short as they quickly divide into a dorsal and ventral primary ramus. Now let's turn our attention to the nerve fibers that are coursing through these various nerves. Let me first orient you to this cross-sectional image. Here we have the dorsal and ventral roots converging to form the spinal nerve, and the spinal nerve bifurcates to form the smaller dorsal ramus and the larger ventral ramus. In total, there are 4 different nerve fibers that course through spinal nerves. General somatic afferent fibers, abbreviated GSA, are neurons that carry information about touch, pain, temperature, vibration, and proprioception from the periphery back to the central nervous system. In other words, these fibers convey sensations from skin, muscles, tendons, and joints back to the central nervous system. The peripheral processes of these fibers can enter through either the dorsal ramus or the ventral ramus. They course through the spinal nerve. And notice how their cell bodies are housed here in the dorsal root ganglion. The central process is then conveyed through the dorsal root back to the dorsal horn of the spinal cord, where the axon, the terminal ends of the axon, will meet up with other neurons in the dorsal horn. General visceral afferent fibers are neurons that carry sensory input from viscera, that is, from organs and blood vessels. They carry that input back to the central nervous system. General visceral afferent fibers typically travel with, but in the opposite direction, of sympathetic and parasympathetic fibers. Shown here in yellow is the course of the general visceral afferent fibers. Notice they can take the same course as the general somatic afferent fibers as they make their way back to the dorsal horn of the spinal cord. General somatic efferents are neurons that carry motor innervation to skeletal voluntary muscles. These cell bodies of general somatic efferents begin within the ventral horn of the spinal cord, and they convey their axons out through the ventral root, through the spinal nerve, and then either to the dorsal ramus or the ventral ramus. From there, they are carried to their target tissues. Lastly, general visceral efferents are neurons that carry motor innervation to smooth muscle, cardiac muscle, and glands. General visceral efferent fibers are further divided into either sympathetic or parasympathetic fibers. Recognize that general somatic efferent neurons begin with their cell bodies located in the lateral horn of the spinal cord. From there, their axons are sent out through the ventral root, through the spinal nerves, and then either course through the dorsal or the ventral ramus. Notice that all the afferent sensory fibers are coursing through the dorsal root, whereas all the efferent motor fibers are coursing out through the ventral root. And lastly, notice that the spinal nerve contains all four fiber types. Along the length of the vertebral column are 31 pairs of spinal nerves that exit the vertebral canal through intervertebral foramina. These intervertebral foramina are usually close to where the nerve originates from the spinal cord. Notice that in the developing embryo at about 8 weeks, the spinal cord extends the entire length of the vertebral canal. Because the vertebral column and dura mater elongate more rapidly than the spinal cord does, the positional relationship between the vertebral level and the spinal cord level does not persist. In other words, the caudal end of the spinal cord in the fetus gradually comes to lie at a higher level relative to the length of the vertebral column. This is why we see the spinal cord ending at the level of L2 in the adult. The level of L2 is considered to be an average level, because in some individuals, the conus medullaris can come to lie at T12 or as low as L3. For our purposes, we'll consider L2 to be the end of the spinal cord, called the conus medullaris. As the vertebral column lengthens, the spinal nerve roots also lengthen, still exiting at their respective vertebral levels where they were initially established in the embryo. Even though this represents S1 at the spinal cord level, notice how this is the S1 spinal nerve exiting just below its respective S1 vertebra. As a review, there are 8 spinal nerves associated with the cervical spinal cord. 12 are associated with the thoracic spinal cord, 5 lumbar, 5 sacral, and 1 coccygeal nerve. All spinal nerves that exit below the level of the T1 vertebra, shown here, will emerge below their respective vertebral levels. So for example, the 9th thoracic spinal nerve will exit below the T9 vertebra. The exception to this rule is in the cervical region. Recognize that there are only 7 cervical vertebrae, however there are 8 cervical spinal nerves. Therefore, cervical nerves C1 through C7 all exit above their respective vertebrae, whereas C8 exits below vertebra C7. This concludes the overview of the nervous system and spinal nerves. Happy studying!